So there's a theory that says that you can't produce high sensitivity and high resolution. So think of sensitivity as how much light is needed to trigger an action potential. Rods are typically very, very sensitive, but they are typically low resolution. So if you think about it at night when you're walking around in the dark, this is good because you want to be able to see kind of the general shapes even in low light, but you don't necessarily need to see very sharp colors. Whereas high resolution is where an image is very, very crisp. And this is going to be the cones. Cones are typically low in sensitivity because they need a lot of input to make them fire, but they give a very high resolution. And part of the reason for this is because the rods show, so these are rods, they show a lot of convergence, which means that we have them coming together to a single ganglion, which is going to go to the optic nerve fiber. So we've got 15 rods here that all converge onto the same ganglion cell. Whereas you can see the cones, so this would be in the fovea centralis. These are 1-to-1, so I've got one cone to one ganglion cell. So this is going to give a lot more resolution because the brain is going to be able to distinguish basically the pixels at all of these different locations, whereas all of this would be one big pixel in a picture. So this would look a little bit grainy. The fovea centralis is where we have the most clear vision, and at that point we have about 4,000 cones and absolutely no rods. And as the image moves away from that fovea centralis, it gets more and more grainy, and you start to see more and more rods and fewer cones. We have color vision, able to span the entire visible light spectrum from purple to red. And this is thanks to our 3 different photopsin cones, which allow us to see color vision. And you can see here, this is where they absorb light, and so their peaks are where they absorb the light the best. Alterations, so mutations, or even lacking one of these cones results in color blindness, and it's usually this green one here, which would lack in the ability to distinguish red from green, which would result in red-green color blindness. This is most common in males because it's on the X chromosome, and so males only have one X chromosome. So about 8% of males in the United States have red-green color blindness, whereas only 0.5% of females are red-green colorblind. Humans also have what's called stereoscopic vision, and that means that the visual field from each of these eyes overlaps here, and it allows for a good sense of depth perception and the ability to judge how far away an object is from the person. And this is something that we see in all of the great apes, including chimpanzees and orangutans and humans, as well as other primates. And this is really important for judging distance. So if you think about organisms that lived in trees that would swing from one branch to the next, you definitely want to know how far that branch is away before you miss it and fall to the ground. Rodents and horses and a lot of other prey-type species tend to have what's called telescopic vision or panoramic vision, where their sight doesn't overlap as much, but they're able to see very wide vision to look for predators or other threats. So take a moment and describe the difference between sensitivity and resolution, and what that means as far as rods and cones.